the topic of the presentation is biochemical sensors for the addictive metabolites. I was mentored by Dr. Shweta. Moving on. First up, these are the table of contents. First, we'll have the introduction. Second, the background check, the methods used, the results we got, and the deliberation upon the results that we got. And finally, I'll be concluding whatever discoveries we made. So first up, we have the aim of the project. So very simply, the aim of the project was to develop a particular biochemical sensor which is able to detect certain addictive metabolites that are present in our bodies, which can be improved with sensitivity, specificity, and predictive potential for addiction monitoring. Right, so it monitors the various addictive metabolites that are there in our body. Now the goal is to detect key addictive metabolites that are present in our body using these biochemical sensors, which is actually the what the entire project is based on. Next is to analyze the addiction related biochemical pathways. There are and third of all, we have to improve early detection, so that we are making this we are using these biochemical sensors so that we are able to discover or detect the particular addiction early on. And fourth of all, to develop a more efficient and accessible addiction monitoring framework. Next up, we have the background on why we chose this topic. First of all, we have the very common rising addiction crisis, right? Now, the substance use disorders and substance addiction continues to escalate year by year. Many people are on opioids, alcohols, other stimulants, cannabis, etc. And the current diagnostic methods often just rely on your behaviors, and they can be a delay, right, because of this. Therefore, there's a need for a biochemical sensor that can detect it early on. Second of all, we have the need for rapid biochemical detection. Like I said, traditional addiction monitoring systems are already very expensive, inaccessible for continuous use over a long period of time. Moreover, we need an early detection and a more affordable option. So, biochemical sensors have the ability to check the fluctuations in our dopamine, cortisol imbalances, and other drug-related metabolites. So this will be even more precise. Third of all, we have the potential of biosensor innovation. There are a lot more advances in the nanotechnology, more variable diagnostic machines, and there has been an acceleration in the biochemical engineering, right? The field of biochemical engineering. Now, this helps to create an opportunity for more portable addiction monitoring systems that can help detect a particular substance use disorder early on, and this can be scalable worldwide. Next, we have the methods that we used to conduct this particular research. First was literature review. We went through existing research on biochemical sensors and addictive metabolites to understand what the current status is about this, and what are the future potential of this. What is the future potential of this topic? Second of all is metabolic pathway analysis. We mapped the pathways which involved dopamine, serotonin, glutamine, nicotine, and ethanol. These were used to analyze, and these were analyzed to identify how these various addictive substances are working on our neurotransmitter systems that can produce certain detectable biomarkers to detect the addiction. Third was the cross reactivity and similarity in analysis. So the sensor specificity was evaluated. The molecular similarity scores and the potential interference of between these metabolites to assess this detection was taken, because in order to see how accurate and reliable these will be in making the particular biochemical sensors. Now, the fourth was AI-based predictive modeling. So a structured workflow was developed to process the behaviors, to process the biosensor outputs, extract relevant features, and apply machine learning techniques for improved relapse prediction and addiction monitoring. Next up, we have the results. First up, we have the better detection of addictive metabolites. So through these, we were able to detect that biochemical sensors will be able to detect important addictive metabolites like nicotine, ethanol, and dopamine faster than other traditional testing methods could. Second, the cross reactivity challenge is found. The study showed that there are some sensors which may confuse similar molecules, which can reduce the accuracy and create false readings. Third of all, stronger understanding of addiction pathways. Metabolic pathways analysis helped to explain how addictive substances affect the brain's chemicals and what sort of biomarkers they can produce, which the biochemical sensors can help to detect. Fourth was the AI improved data analysis. So, using the AI workflow, it helped to organize the biosensor data more effectively, and this made prediction of relapse and monitoring of the particular addiction much more efficient. So, first of all, here we have the AI workflow, which I explained, and how it helps in the relapse prediction. Next up, this was the cross reactivity heat map, which we saw, which will basically help to detect whether the biochemical sensor will be able to particularly does not mess up the detection and is accurate. This was the metabolite pathway integration strength, the interaction intensity, which is required again for making sure that the particular biochemical sensor is accurate in its detection. Now, this is an explanation of the previous slide. The molecular docking results showed different binding strengths between the tested addictive metabolites and the target protein receptor. So, catecholamine had the strongest binding affinity, indicating the most stable interaction with the receptor. Nicotine showed moderate binding strength with ethanol, and ethanol had the weakest binding affinity. So these results particularly suggest that the catecholamine related metabolites are much easier to detect because they interact more strongly with the addiction related receptors. Nicotine is mildly will be lesser, but it also showed an important receptor interaction, whereas ethanol had weaker binding. It may interact less directly. Overall, the docking analysis supports the idea that different addictive metabolites vary with the biological interactions with the receptors, which is actually very important for when you're designing a biochemical sensor. So stronger binding metabolites may provide more reliable detection targets, improving the sensor sensitivity, and will help in a better prediction. Now, these are the results of the mapping. This diagram shows how addictive substances such as ethanol and nicotine affect the brain's reward system. So they interact with some of the very important neurotransmitters like dopamine, serotonin, glutamine, and catecholamines, which make you happy. So dopamine plays a major role in pleasure, motivation, and reinforcement, which is actually key towards the development of an addiction. That's why people take these substances. Nicotine and ethanol can disrupt the normal neurotransmitting signaling, which increases the amount of dopamine that is being released in our body, giving us that euphoric high. The image also highlights how multiple biochemical sensor pathways are interconnected with each other, showing that addiction is not only caused by a single chemical, but actually a very complex interaction within the nervous system. And therefore, understanding these pathways is important for developing a particular biochemical sensors. Here are the various other roadmaps that we studied. Now, from this, basically, we got the pathway analysis showed that the addictive substances like nicotine and ethanol affect many important systems in our body. Not just one chemical pathway, as we saw, it had an interaction between many chemical pathways, and that's why it is so complicated. The tyrosine metabolism roadmap explained how dopamine is produced and how it is connected to the addiction, reward, and pleasure the system of our body. Since dopamine is strongly linked to motivation, the changes in the pathway can increase the risk of this addiction. The tryptophan metabolism pathway showed that serotonin is also involved. This means addiction can impact your mood, emotions, mental balance, and not just physical dependence, because that's what serotonin is involved with. So the Theon metabolism pathway highlighted how the body responds to the various harmful substances through detoxification. This suggests that addiction can create stress at a cellular level, which may affect you in a longer term. The xenobiotic metabolism pathway showed how the body breaks down foreign substances like nicotine and ethanol into metabolites. These metabolites are what are actually important because these are what the biochemical sensors can detect while monitoring for an addiction. Overall, the results suggest that addiction is a complex biochemical process, right? It involves multiple neurotransmitter, as we saw, multiple metabolic pathways, and detoxification systems. So this supports that the importance of using biochemical sensors and advanced data analysis can help actually in addiction detection and monitoring. Concluding this with, this project basically showed that addiction is complicated, involves multiple neurotransmitters. Substances like nicotine and ethanol can affect various important brain chemicals like dopamine, serotonin, glutamine, which affects our reward system, mood system, and makes us dependent on these particular substances. Now, biochemical sensors have a very strong potential to detect these addictive metabolites quickly and efficiently than the current traditional methods that we have. So, by combining the pathways analysis with AI-based data processing, this research also demonstrated how technology, the current present technology, can help in improving detection, addiction monitoring, and relapse prediction.